acquired immunity, also known as adaptive immunity. The resistance acquired by an individual during life, it's a subsystem of the immune system composed of cells and processes that eliminate pathogen by preventing their growth. It is of two types, active and passive. Active immunity, the immunity which results from the production of antibodies by the immune system in response to the presence of antigen, any microorganism. Passive immunity, when a person is given antibodies to a disease rather than producing them through his or her own immune system. That is called as passive immunity. Active immunity involves in functioning of host immune apparatus like synthesis of antibodies and production of immunologically active cells. That is subdivided in two types, natural and artificial. Natural through clinical or subclinical infection and artificial induced by vaccination. Active immunity develops humoral and cellular immunity. Latent period. It's a period between infection with the microorganism and the onset of symptoms. For example, between exposure to radiation and the appearance of cancer. That time period called as latent period. Next term is negative phase. It's a time period in which the level of measurable immunity is reduced that may follow the injection of foreign antigen in active immunization. It occurs when the person is exposed to the same antigen second times. At this point, immunological memory has been established and the immune system can start making antibodies immediately. That is in case of second response. Now, humoral immunity. It is mediated by macromolecule formed in extracellular fluids such as secreted antibodies, complement proteins, and antimicrobial peptides. That is named humoral immunity because it involves substance found in the humors or body fluids. Humoral immunity neutralize toxin of antigen, remove antigen by phagocytosis, and cause lysis of antigen. CMI, cell mediated immunity. CMI is an immune response that does not involve antibodies. Rather, CMI is the activation of phagocytes, antigen specific cytotoxic T lymphocytes, and the release of various cytokines in response to antigen, any microorganism. That is called as cell mediated immunity, CMI. Natural active immunity. It is acquired by natural subclinical or clinical infections. Such immunity is long lasting and varies on types of pathogen. It occurs when the person is exposed to a live pathogen, develops the disease and become immune as the result of primary immune response. For example, active immunity against poliovirus and lifelong immunity against chickenpox, and also influenza and common cold. They are the examples in case of natural active immunity. That is less permanent in bacterial infections than viral infections. The term premonition, also known as infection immunity, it is a host response that protect against high number of parasites and illness without eliminating the infection. This type of immunity is rapid, short-lived, and partially effective. Artificial active immunity. Active artificially acquired immunity refers to any immunization with an antigen. It's a resistance induced by the vaccines. Vaccines prepared from live, attenuated, or killed microorganisms. These are two different categories, or also prepared from their antigen or toxoids. In killed vaccines, the organisms are killed by heat, formalin, phenol, and alcohol, and then preserved. The next term, toxoids, these are prepared from bacterial endotoxin inactivated by formalin or alum. Next term is booster dose. Booster dose is extra administration of vaccine after an earlier dose, needed periodically to boost immune system. For example, shots of tetanus and diphtheria is recommended for adults every 10 years. Live vaccines replicate in the host, causing a mild or undetected disease and bring about an immune response which mimic natural infection. It parallels with natural but lower grade. For example, BCG, MMR, Sabin vaccine for polio. Next is killed vaccines. It's an inactivated vaccine consisting of virus particle or bacterial particle or other pathogens that have been grown in culture. It's comparatively less immunogenic. Protection lasts only for a short period and supplemented by booster dose. And by adding adjuvants, we can improve antibodies response. In the diagram, you can see live attenuated killed pathogens and different microbial products used in production of different bacterial and viral vaccines. In this diagram, you can see different available vaccine against pneumonia, tetanus, diphtheria, and meningitis.